speaker this morning is Ian Lowe from Denison University, and the title of his presentation is A Mechanochemical Model for Myosin 6.
All right. As he said, I'm Ian Lowe. I work with Dr. Trevor at Denison University in the Computational Biophysics Lab. So today we're going to go over what is a protein, what specifically is myosin 6, why do we care about it, the model that we built, the computations and how we analyze all of our data, and then how we compare our data with actual myosins walking around in cells. So what is a myosin? First of all, it's a protein, which is a polymerized amino acid chain. That's actually myosin 6, drawn as a cartoon. It's walking on its little actin filament. That's how it steps, hand over hand. There's a bunch of different purposes for proteins. Some of them are used in synthesis of other proteins, helping other proteins fold. There's myosins and kinesins and dyneins that are transport proteins, so they bring things around your cell. And then myosin 6, as I said, is an intracellular transport protein. It brings things, mostly little vacuoles around your cell. And then dimerizes, so each of these is a single myosin 6 to form a myosin 6 that's two proteins wrapped together so that it can walk processively. So as I was saying, it's a transport protein, but it's a very unique one because it walks in the opposite direction of all the other myosins. All of your other myosins walk from the inside of your cell to the outside. This one, for some reason, we don't know, walks from outside in. And we think it has to do with how this lever arm is attached to the motor domain. And there are multiple gating mechanisms that cause it to have this processive motion, which means why does it walk in one direction if it's just a stochastic, random walking protein? It's got to be from some sort of bias to how it's diffusing, how it's pointing at any given time. And that's where these lever arms come into play. So there's two different competing theories that have to do with the stiffness. There's this theory, which says this little blue area right here unfolds into these really, really floppy blue areas. And then there's this theory, which says that little blue area, which is analogous to that blue area, doesn't unfold. It's much stiffer, and you get this green area, which would be up here, using to extend its arms. They both give a 36 nanometer step size, which is the pseudo-repeat of actin, and they both walk at about 148 nanometers per second. So we built this model. For each head, you have a simple three-state model. You have a waiting empty state, then you have an ATP-bound state, which immediately hydrolyzes ADP-PI. That's basically the fueled state. And then the ADP-bound state, which is the waste-filled state. So a simple walking would be blue to green to black, and then repeating that, and that would be it stepping. But you don't always get that. You get empty being filled up with waste and dumping waste over and over again, just because it's diffusing into there. So we had to include these waiting pathways. And then we took two heads because you always have a dimerized protein that's walking, and we get this really messy model, but it elegantly displays walking and waiting pathways. A simple walking pathway would be from this state, down this blue line here, over here. This green line implies a forward step as your T in the back goes to a D in the front, and you get walking, forward walking rather. And then this state would be a backward step, and if you want a processive protein, so you want one moving forward, you hope that this happens a lot less than this. And so the way we model this actually is just a series of a couple differential linear equations, first order, and each of these represents the rate of change for each of these states. You solve these, mathematics is great, And with all these rate constants, most of those will come from literature, but two of them, which have to do with diffusion, you can't actually experimentally measure because of how tiny these things are. So we use the combination of polymer bending simulations and CPU diffusion simulations to simulate them. So this is our model. You have those little heads that I was showing you right here, and then this will be the arm. The green part will be the arms, and the polymerized right here. And from that you can get rates for these. And because of how fast they are relative to the rest of the reaction, an estimate is about all you need for these, so about an order of magnitude. The ADP, so the waste dumping and re-diffusing into it, is actually the rate limiting step. So we're happy with this estimate for now. And using computations, so we solve these master equations, you get a steady state system, you get your probabilities of being in each state, we normalize them. So they're just arbitrary probabilities. You can calculate different system values. So I was talking about how it walks, and right now we're working on velocity calculations. So you have a forward velocity calculated from the forward diffusion times the number of states that are able to walk forward, and the backwards velocity from the diffusion constant, that's the number of states that are able to walk backwards. And then your net velocity is going to be your forward minus your backwards. And that's what we would like to see matching experimental data so that we know our model works. So as I said, it's dependent on your steady state concentrations, rates, and then we look at how the concentration of ADP and ATP, as well as these two diffusion rates here, affects the velocity. And I was talking about the diffusion rates. This is actually the diffusion bias, which goes into the rates, saying that your forward diffusion is 100 times more likely than your backwards diffusion because of the shape of the protein. With these persistence lengths, which means how stiff the protein is, which is what I was talking about with the two competing models, you get the same velocity. Using forward diffusion bias at tenfold, however, you still get the same velocity. So we know that our model is fairly robust in terms of persistence lengths, but there is still a definite change with diffusion bias. So we wanted to add this next sensitivity analysis on ADP, and you actually look at gating, which is one of the three mechanisms I mentioned earlier. So it's a multiplicative change to favor how the polymerized protein picks up ADP. And so here would be the experimental measure of the velocity. It's about 140 nanometers per second. And if you tell the forward head to take out less ADP, you get a lower velocity, because that means your trailing head, which is the one that lets you step forward, is gunked up with waste. But if you tell your trailing head to take up less ADP, the velocity goes up because it doesn't get gunked up with waste. So, I guess in conclusion, we've been able to work or capture out the kinetics fairly accurately within our differential equation framework. Just using the diffusion gating parameter, our data is about 5% higher than experimental velocity. When you use the ADP gating parameter and the diffusion bias, we accurately put experimental values. And then, so talking about the competing lever arm models, we found that both are viable for our model, but in the future, we may be able to differentiate between them. And going forward, I talked about ADP and diffusion gating, but we also want to look at ATP gating, so how it uptakes fuel. We also want to get more data on polymer diffusion, because we'd like smaller polymer beads to get better persistence length measurements. And we also want to look at more quantities. So we looked at velocity, but we also want to look at run length, which is how long it actually stays on the actin before it falls off, and duty ratio, which is how long it spends totally bound in the waiting or wasteful states compared to how long it's diffusing. And that's it.
Questions for Ian? Yeah.
So you showed us a beautiful set of coupled first order applications, and then you said that they have a steady state solution. Is
Yeah.
there a way that we can see that? You convince me that other
It does. I don't actually have a picture of it because it's dependent on how we change the rate constants these two. So it has enough like boundary conditions that you can solve that.
Is it? But do you? Maybe a better question is: if you change the parameters, does it become unstable? Is it stiff or?
Not that I've seen.
So, sort of thinking on a grand scale, does this work inform us at all for development of pharmaceuticals or like nano machines or anything like that?
This, for the, in theory, you could look at pharmaceuticals, but I think it's more important to like nanoscale machines because these are thought to be force gated. So you have some sort of tension force because these are bound and being pulled back. It's not exactly a just limp lever arm. So we're trying to figure out why we can get something that is just diffusion to walk in consistently in one direction. Does that answer your question?
Yeah, I think so. I mean, is there work going on right now trying to take sort of our understanding of the mobility of these myosin proteins and actually make those sorts of nano machines, or?
I don't have it. It's a little bit outside of my scope right now.
Okay, any other questions? Okay, now let's thank Ian again.